Fine. Now let's start and not waste any time. This is the March 2024 paper one exam. Okay. Question number one. A student used a light microscope to observe a blood smear on a microscope slide. An eyepiece graticule was used to measure the diameter of the white blood cell, and the student recorded that the white blood cell was 5 eyepiece graticule units in diameter. Which additional information does the student need to determine the diameter of the white blood cell in micrometers? A. Calibration of the eyepiece graticule using a stage micrometer only, or calibration of the eyepiece graticule using a stage micrometer and the magnification of the lens, the magnification of the eyepiece lens only, or the magnification of the eyepiece lens and the magnification of the objective lens. Which one of those do we need in order to measure the actual diameter of the specimen?
B.
Farouk.
Yes.
Here we're using an eyepiece graticule and a stage micrometer. Do you know what is an eyepiece graticule?
Yeah.
Yeah, the
Uh,
ruler that is found
yes,
on
the
the
rule.
eyepiece itself. Yes.
Yes.
And the stage micrometer, this is the slide that has a ruler. Remember when I draw the ruler on the slide? And the ruler on this slide, it has a scale. We know the length of this ruler, but we don't know the length of the ruler on the eyepiece graticule. It depends on the magnification. Now, I have a specimen and I want to know its actual diameter. What do I do? Or what do I need? I need to do something called calibration.
Yes.
I think you know what is calibration, in which
Yes.
the
Right.
ruler of the eyepiece graticule will be superimposed on the ruler on the stage micrometer, and they will be both brought on top of each other, and they will be both in focus. And I will know how one division, the value of one division on the eyepiece graticule, how much does it measure on the stage micrometer? Don't we see pictures with the stage micrometer ruler, a bigger ruler, and a smaller ruler, which is the eyepiece graticule with 100 divisions? Okay. And then let's say those 100 divisions, they occupy 2 divisions of the stage micrometer. If every division is 1 millimeter, then those 100 divisions on the eyepiece graticule is equivalent to 2 millimeters on the stage micrometer. Then one division on the eyepiece graticule is equal to 1 times 2 divided by 10 millimeters on the stage micrometer. Do you know all these steps?
Yes, I do.
Okay, so in order to carry out those steps, what do we need? What are the things that we need in order to find the actual length of a specimen? Because after I finish calculating or after I finish calibration, I will remove my stage micrometer and I will put the specimen. And the specimen will be under my microscope, and the ruler of the eyepiece graticule will be on top of it. And I'm going to see this specimen occupies how many divisions on the eyepiece graticule. If let's say the specimen occupies 10 divisions on the eyepiece graticule, I will multiply by 10 by the value that I got in the first step. So I explained all the steps in detail. Now, what is the information needed by the student to determine the diameter of the specimen? What are the things that I need to know the actual diameter of a specimen using my microscope?
A.
Yes, it's A. You gave me the right answer only when you understood the question. I need the calibration. I need the eyepiece graticule, and I need the stage micrometer. The magnification will be adjusted, and I will not change the magnification. I need those 2 things, and I will have to bring them in focus and determine the diameter of one division on the eyepiece graticule using my stage micrometer. And then I will remove the stage micrometer, and then I'm going to put my specimen so that I can measure the actual diameter of my specimen. So is question one understood?
Yes.
Okay, good. One is A. Now let's read two. Which statement explains why it is necessary to use an electron microscope to see the crista of a mitochondrion? The magnification of the electron microscope is greater than that of the light microscope, or the membranes of the crista are separated by a distance that is greater than 200 nanometers, or the maximum resolution of a microscope using visible light is too low. The wavelength of an electron beam is longer than the wavelength of visible light. First, Farouk, tell me, question number 2. Which answer are you sure it is incorrect? A, B, C, or
Right.
D? Excuse me?
1 second. D.
Yes. One of the easy ways to answer multiple choice questions is to answer by exclusion. I'm going to remove what I am sure is incorrect. So D is incorrect. Two
Yes.
more are incorrect. Let's look at A. The magnification of the electron microscope is greater than that of the light microscope. A is right, but we
Yes.
are here looking at the crista of the mitochondria. The crista, these are the inner membranes. These are the crista of the mitochondria. Okay, this is the mitochondria and the crystal. Now, these are very, very small details. I have an inner membrane and an outer membrane, and the distance between them, if the distance is too small, I will not be able to see it with a light microscope. Now, in order to be able to distinguish between two separate pictures or two separate structures, is the magnification important here or the resolution?
The resolution.
The resolution. So A is wrong, because the magnification is how many times bigger the object is to the real thing. We are looking at the resolution because we want to look at a detailed structure, and the resolution here makes you see details. B, the membranes of the crystal are separated by a distance that is greater than 200 nanometers. Is
No.
this correct?
No. Less.
Excuse me?
It's separated by less than 200 nanometers.
Yes, you are correct. So B is also wrong. Because if it was separated by a distance that is greater than 200 nanometers, I can use it. I can see it using a light microscope. So what is the correct answer for number 2?
If my I got disconnected for a second.
What is the only answer left then?
C.
Yes, okay. The resolution of a light microscope is very low, and I cannot use a light microscope to see details. So 2C. Okay. 3. Some stains can be used to identify cell structures. In a dilute solution of one stain causes the whole cell to appear blue. The blue color rapidly disappears from most cell structures, and those cell structures that release energy stay blue. So which type of cell structure is likely to stay blue? Which cell structure
The
of those releases energy? Yeah, this is very easy.
mitochondria.
Yeah, okay. 3 is very easy. 4. When mucus is secreted from a goblet cell, these events take place. 1. Addition of carbohydrate to protein, fusion of a vesicle with the cell surface membrane, extracellular release of a glycoprotein, separation of a vesicle from the Golgi body. What is the sequence in which these events take place? What's the first thing that happens?
1, then 4, then 2, and then 3.
You are correct. It is A. Because first we will do the modification, and then the vesicle will be separated from the Golgi body, and then it will bind with the cell surface membrane, and it will be released. Perfect. 5. The diagram shows 4 biological features. We have DNA, capsid, 70 Vedberg ribosomes, and RNA. Which biological features are present in typical prokaryotes?
The 70 ribosomes
Excuse
and
me? The
pro-
70 Vedberg ribosomes,
yeah,
yes.
the RNA,
Okay, yes. Bacteria can have any type of genetic material, RNA or
the DNA.
yes.
DNA
Does
or
bacteria have capsid?
no, that's virus.
Yes, exactly. So it's 1, 4, and 3. So it's C. 4, 5, C. Good. 6. Which row shows features that occur in dicot plant cells and also in typical bacterial cells? Checkpoint means it is present and cross means it's not present. Okay, now which one of those is present in plant cells and in bacterial cells? Choose A, B, C, or D.
Okay. The 70s ribosomes, yes, because it's in both.
Yes,
The 80s
correct.
ribosomes, no.
So C is wrong. D, those two cannot be right. It's either A or B. Which one?
B, because there's circular DNA in both.
Yes, not only that, the centrioles, they're not found in bacteria or plant cells. Centrioles are only found in animal cells.
Yeah.
So it's
I was
B.
I wasn't sure about that one, so I just went to the DNA because I knew it was correct.
Well, it is correct. 6 is B. Which flowchart outlining the test for non-reducing sugars is correct? Don't forget non-reducing sugars like the sucrose. A, we add alkaline sodium hydrogen carbonate to the sample first, or
No,
we boil the sample with dilute hydrochloric acid.
boil it first.
Yes, so this means we will exclude A and B because the first step is wrong. So the answer is either C or D. Let's look at C and D. We will boil the sample with dilute hydrochloric acid, and then we will neutralize with sodium hydrogen carbonate. So the first two steps are the same, and they are correct. Now, the last step here will make the difference. Either we add Benedict and boil, or we add burette and boil.
Benedict.
What's the test for non-reducible? Easy. This is easy. 7C. 8. Okay. Which diagrams show the release of a water molecule during the formation of a glycosidic bond? Look at one. You have two hydroxyl groups
Mm,
and they will join releasing water. And look at two. You also have two hydroxyl groups and they will join releasing water. And look at three. You have also
all of them.
two, all of them. Yes, all of them. 8 is A. Okay. Here, this is a branching um, amylopectin 1 to 6. Now, which molecules have a structural formula that contain C double bond O bond, the carboxylic acid, amino acid, fatty acids, glycerol, or protein? Now, here you need to remember the structure of the molecules that we studied.
Yeah. So the amino acids have
Yes, correct.
the fatty acids too as well.
Yes, true.
Okay. And the protein thing.
Yes, correct.
Is the
Perfect.
amino acid, yeah.
Not
Okay.
the glycerol. The glycerol is a polymer made up of alpha glucose, and the alpha glucose, these are rings and they do not have a carboxylic acid group. Excellent. If we want to remember the structure of the amino acid, we have a carbon and an amine group, NH2, and another carbon bonded to a carboxylic acid, correct? Hydrogen, and the variable R group. This is an amino acid. If we want to remember the structure of fatty acids, we have a hydrocarbon chain with a methyl group at one end, CH3, and also a carboxylic acid group at the other end. And this is a hydrocarbon chain. Okay, good. So, of course, proteins are made up from amino acids. So it's 1 and 2 and 4, which is B, correct. Now, 10. Which statement about triglycerides is correct? In any triglyceride, all the fatty acids are saturated or all are unsaturated. B. Two triglycerides are joined together by an ester bond. C. Triglycerides are polar hydrophobic molecules. D. Glycerol is joined to each fatty acid by a covalent bond. Now, this is also easy. Choose the incorrect answers and you will have only one correct answer left.
So C is wrong.
Correct. C is definitely wrong, so I will exclude it.
And B is as well.
B is wrong, of course, because it's a triglyceride, not two. So B is wrong.
And A is incorrect because it can be, yeah. Okay.
Yes, of course, A is wrong. The answer is D because ester bonds are covalent bonds. Very good. 11. Many flowers produce a sweet solution called nectar. Bees provided with nectar use enzyme Q to change the nectar into honey. After testing a sample of nectar for the presence of reducing sugar using standard lab reagents, the sample was blue. After testing a sample of honey in the same way, the sample was orange. Which conclusion about the reaction catalyzed by the enzyme is consistent with these results? So pay attention to this sentence. We tested a sample of nectar okay, for the presence of reducing sugar using standard lab reagents, they mean Benedict, and the sample was blue. So does the nectar have any reducing sugar?
And yeah,
The
no.
result was blue.
What?
No, okay. Now, after testing a sample of the honey in the same way, now they tested honey for reducing sugar, and the result was orange. So does honey have reducing sugar?
Yes.
Yes. Good. Which conclusion about the reaction catalyzed by the enzyme is consistent with these results? So, what is the type of the reaction, and what is the substrate, and what is the product?
It's okay. It's hydrolysis because
Excellent.
it breaks down
Because we have to add
protein.
water to break the bond. Yes.
Okay, and then the substrate is maltose is only in milk, right?
Maltose is only what? Excuse me?
Isn't maltose only in milk? No, that's lactose. Sorry.
Number one, yeah,
Yeah,
it's
sorry.
not in milk. And also, maltose, what is the only non reducing sugar? Because we know that the nectar is non reducing, sucrose. Yes. After we broke down the nectar using the enzyme into honey, because they said that the nectar was broken down by the enzyme into honey. So we had nectar, it was broken down by enzyme Q into honey. This enzyme did a hydrolysis reaction. It broke down the nectar, which is a non reducing sugar, okay, which is definitely sucrose. This is the only non reducing sugar actually that we studied into reducing sugars. Sucrose is glucose and fructose. So what's the answer?
C.
C. Okay. This is all biological molecules. Again, biological molecules. The diagram shows the structure of part of a peptidoglycan molecule. Peptidoglycan means this molecule is made up of what, Farouk?
Uh, amino acids.
Peptido means a protein is involved. Yes, you are correct. And glycan means a carbohydrate is involved. So here you have a molecule made up of part proteins and part carbohydrates. Okay, now this structure, by the way, it should be okay with you. What is this structure here? And what are these?
The top is
Hmm,
uh alpha alpha beta glucose.
excellent.
Yeah.
Excellent. This is beta glucose. Well done. We knew it from carbon 1.
Yeah.
Hydrogen is below the ring. Very good. And this part of the molecule is
Amino acids
excellent. How did you know?
because of the NH.
Because of the nitrogen. Well done. Now let's see the question. Which type of 1 to 4 linkage and how many peptide bonds are shown in this part of the molecule? Now, how many peptide bonds are in this molecule?
Okay. Let's see. One, two, two,
Mhm.
three.
Why 3? Here is 1, here is
Yeah,
2, here is 3,
four.
because the peptide bond, yes, is 4.
I
So
think
we will go to the answer is either B or D. Okay?
B
We
because
will
B.
exclude A and we will exclude C. Now, is it B or D? Let's go
D.
and look at the type of linkage. What did you say?
It's D because beta. It's beta glucose.
Correct. Yes. Well done. So you just need to be um like notice those small things and you'll get the answer. This is beta glucose. Good. So the answer is D. It's not B. Well done. 13. A scientist investigated the progress of two enzyme catalyzed reactions in separate test tubes, X and Y. Both reactions result in color changes that can be detected using colorimetry. Zero point 0.5 centimeter cube samples were taken from each test tube at the start of the investigation and at regular intervals for the next 5 minutes. Copper ions were added to each sample as soon as the sample was collected to inactivate the enzymes and stop the reaction from progressing further. The absorbency of each sample was measured using a colorimeter. The graph shows the results. Now, when you have any graph, you look at the and the y-axis. Here, the time is on the x-axis and the percentage absorbency is on the y-axis. Now tell me, Farouk, if the solution is dark in color, it will have a high absorbency or a low absorbency?
High absorbency.
Very good. Which statement is consistent with the results shown in the graph? Let's look at A. The substrate in test tube X has a higher absorbency than the product. B. The product in test tube Y, the product in test tube Y has a lower absorbency than the substrate. Always the substrate is at the beginning and the product is at the end. C. The rate of the reaction in test tube X increased with time. The rate of the reaction in test tube Y increased with time. So what is the most correct answer? The most obvious one. This is which number? Yeah,
B.
correct. Perfect. The
Yeah.
product in test tube Y has a lower absorbency, yes, than the substrate. Good. This is the substrate, and here at the end of the reaction, we have the product. So it's B. Well done, Farouk. 14. A student investigated the hydrolysis of lipid and high-fat milk using the enzyme lipase. Now, tell me, Farouk, when lipids are broken down, what do they give you? What are the products?
Glycerol and fatty acid. One
Good.
glycerol and fatty acid.
Okay, put this in mind because fatty acids are acidic. 1 centimeter cubed of enzyme solution was added to the 10 centimeter cubed of high-fat milk. The temperature was kept constant and the pH of the reaction mixture was recorded at time 0 and every minute for 20 minutes. Which statements correctly describe the expected results of this investigation? 1. The product forms more slowly as time proceeds because the concentration of the substrate is decreasing. Let's take it one by one. Is sentence number 1 correct? The product forms slowly as the time proceeds because the concentration of the substrate is decreasing. Is one right or wrong?
It's correct. I think
Yes,
so.
of course, it's correct. Well done. Always the initial rate of the reaction is highest because the substrate is present in excess initially, and with time the substrate decreases. Correct. So 1 is right. 2, the pH of the reaction mixture increases rapidly in the first few minutes and then increases less rapidly. Is 2 right or wrong?
No.
Correct. Well done, Farouk. 3, because the products are fatty acids and glycerol, and fatty acids will lower the pH, not increase the pH. 3, the increase in the concentration of product eventually causes the lipase molecules to denature. Is this right or wrong?
Yes, because
Excellent.
they decrease the
Well
pH.
done. Yes. So the answer is
C.
well done. Amazing. This is a very good job. 15. What is the correct range of measurements for the width of the cell surface membrane?
Is B.
Excuse me?
No, A.
No, it's B.
B. Okay.
Go back to your notes. It's a 7 nanometers. So it's somewhere
Yeah.
between 5 to 10 nanometers. Okay, 16. Where in the cell surface membrane are the carbohydrate chains of glycoproteins and glycolipids mainly located? Remember the glycoproteins and the glycolipids?
Yes.
Okay. You know, we have peripheral proteins and transmembrane proteins. If this is the cell membrane, these are the phospholipids. We're not concerned with them now. We have proteins and then a carbohydrate chain. This is the glycoprotein. And if we have, no, we are concerned by the lipids, by the phospholipids. Phospholipids, we have here a carbohydrate chain. It will be a glycolipid. So where are they located? On the outer surface of the membrane or on the inner surface of the membrane?
On the outer surface.
Yes. Outer surface. They are both on the outer surface. Sodium ions can enter cells across the cell surface membrane. Which methods could be used by sodium ions to cross a cell surface membrane and enter a cell? Active transport only, active transport and facilitated diffusion and simple diffusion, or simple diffusion only. Give me an answer and
Act-
tell me why.
Active transport and facilitated diffusion. because they're polar
Well
and
done.
hydrophobic core.
Yes. Simple diffusion is incorrect because they're polar and they have to travel or cross the membrane using some sort of channel or carrier protein. Perfect. So 17 is B. The diagram shows how an artificially partially permeable membrane was used to separate a 5% sodium chloride solution and a 10% sodium chloride solution in a beaker. The two sides of the beaker were labeled R and S. So you have here a partially permeable membrane, and you have side R and side S. Side S is 10% sodium chloride solution. Side R is only 5% sodium chloride solution. Which row correctly describes and explains what will happen in the half of the beaker labeled S? So we want to know what will happen here. Let's see. Description of S. Volume of the solution will increase or volume of solution will decrease. First, I want to choose A, B,
Okay,
C, or D. The volume of S will increase or decrease.
increase because the water will move
Of
in.
course, it will increase. You are
Yeah.
correct. So I will exclude C and D. Now, is it A or B?
From low water, wait, A, from high water potential to low water potential.
Well done. Perfect. It is A. Okay. Agar cubes can be used to demonstrate the effect. All this is lesson cell membranes. Agar cubes can be used to demonstrate the effect on diffusion of changing the surface area to volume ratio. Three different agar cubes made using a colored indicator solution were placed into dilute hydrochloric acid. Dilute acid that diffused into the cubes. As the acid diffused into the agar cubes, the color of the indicator solution changed. The cubes had volumes of 1 centimeter cubed, 2 centimeter cubed, and 3, and were left in the dilute acid for 10 minutes. All other variables were kept the same. After 10 minutes, the agar cubes were removed from the dilute acid and cut in half. The cut surfaces were observed, and the results were recorded as diagrams. All diagrams were shown to the same scale. The results for the 2 centimeter cube are shown. So this is the original color, and this is the color change that happened after they were left in the dilute acid for 20 minutes. Now, do you understand what is the experiment about, Farouk? Do you understand what's happening?
Not really, no. I'm confused.
We have this experiment in the lesson cell membrane. You will, if you are confused, then do not proceed with the question, except when you go back and read it again and understand it. I'll explain. There were three agar cubes, okay, of different surface areas. And the three of them were colored. In the notes, they were pink, okay? They were colored. And then they were put in dilute acid for 20 minutes. This acid will diffuse into the agar cube. And as the acid diffuses into the agar cube, the color will change. The color of the cube will change from pink to colorless. This is the traditional investigation. Now, here we are testing surface area to volume ratio. Of course, when the cubes have a high surface area, less time will be needed for the agar cube to completely become colorless, or in the same amount of time, more or most of the cube will become colorless. Do you understand now the investigation? Do you understand what I said?
Yeah.
Okay, good. So here they gave you a scale for the 2 centimeter cube after 10 minutes. Fine. After 10 minutes, some of the acid diffused, and it made some of the agar colorless, but not the whole cube. So it needed more time for all the color to disappear, but we only kept it for 10 minutes. Now, which diagram show the results for the 1 centimeter cubed and the 3 centimeter cubes?
Wait.
This
So
is which number? Which one had the biggest or the largest surface area? The 1
three, no,
or the
three,
2 or the 3?
the three.
No, you're wrong, Farouk. The
Wait.
3 centimeter cubed is the volume. So this means high volume and low surface area. And the 1 centimeter cube is also the volume.
Mm-hmm.
So We are looking at a cube with high surface area to volume ratio. So the higher the surface area, the faster the diffusion. And the smaller the surface area, the slower the diffusion. Here, they gave you the size 1 centimeter cubed and 3 centimeter cubed. It's obvious that the 1 centimeter cubed is smaller with a large surface area. Yes?
Yes.
Small cube, this small, this size. Now compare the 1 centimeter cubed. This is in terms of volume. Okay, it has a small volume, and this has 3 centimeter cubed. This has a bigger volume, but also a smaller surface area in relation to volume. If you have a smaller volume, you will have a larger surface area in relation to volume. When we came to study this concept, we said if you have an animal that is unicellular, it has a small surface area and a small volume, but the surface area compared to the volume will be much bigger. And if you have a multicellular organism like an elephant, remember our elephant? Okay, this elephant is growing and growing and growing. As the elephant is growing, both the surface area and the volume are growing, but the volume grows much faster than the surface area. So this elephant has a smaller surface area to volume ratio. That is why this elephant needs a respiratory system and a circulatory system. Remember this concept, Farouk?
Yes.
Okay, but unicellular organisms, they have a small surface area, yes, and a small volume, yes, only 1 centimeter cubed, for example, but the surface area to volume ratio is very high. In other words, the surface area is very high compared to the volume. They're both small, but the surface area relative to the volume is much higher. So they don't need a circulatory system or a respiratory system, and they can exchange materials with their surroundings simply by diffusion. Diffusion is efficient and it is good enough. Okay. So the 1 centimeter cube is a cube of high or low surface area.
High surface area.
Very good. High surface area. And the 3 centimeter cube is of a high or a low surface area.
Low.
Low surface area. Good. It has a big volume, yes, but a low surface area. Now we left both cubes for 10 minutes only. Which one will show a bigger area of discoloration? The one or the three?
The one.
Of course, because I have a very small surface area. This means that the acid will diffuse faster. The faster the diffusion of the acid, the greater the discoloration. Yes? We left it for only 10 minutes. So after 10 minutes, after I get out the cube and I cut it and I look inside, how will the 1 centimeter cube look like? And how will the 3 centimeter cube look like?
See.
Yes, well done. Okay, now go back and revise this experiment in the lesson cell membranes. You'll
Okay.
find it. It's taking a whole page. I know. Okay, yes, you are right. It is C. Fine. So 20, we are halfway through. During sperm formation in mammals, part of the structure of each chromosome is replaced with proteins called proteins. This replacement allows the DNA to be packaged much more densely than would otherwise be possible. Which part of the chromosome is replaced by proteins? In other words, what in the DNA, which structure in the DNA helps in the packaging and the dense packaging of
Extone.
the DNA? Yes. Okay, they're just playing around with words. Okay. The diagram shows the mitotic cell cycle, during which phase do chromosomes condense and become visible? A or B or C or D? And they're telling you that this small part here is cytokinesis.
Okay. So condense and become visible.
Mm,
It's a prophase.
correct.
So A in the interface.
Farouk, you're wrong.
Oh no no, sorry.
Mm,
Okay, I'm sorry. The chromosomes condense and become visible.
mm.
The prophase. So.
Yeah,
So
the
A,
prophase is part of what? It's not A at all.
yes, A is the interface.
A, B, C, the
Yeah.
three are interface. This is G1,
Oh,
this
okay.
is synthesis,
Oh,
and this
okay.
is
I
G2.
got
And what is D?
That's mitosis. Okay, it's D. Sorry.
Yes, it's
Yes.
the prophase and mitosis. Good. So here condensation happens in prophase. Okay, be careful. Now, the last 10 questions or the last 20 are usually harder. Which statements correctly describe features of stem cells that are essential for their role in cell replacement and tissue repair? So we want to choose the correct sentence. Now, in a question like this, it's easier to choose the incorrect. So we exclude the incorrect. So let's look for incorrect sentences. After mitosis of stem cells, the daughter cells can either remain a stem cell or follow a developmental pathway that leads to the formation of specialized cells.
Yes.
Is one right or wrong?
Right.
It's very right. One is definitely right. Okay. Two, stem cells are different to all other body cells because they retain all of the genetic information in their DNA throughout the life of the organism. Is two right or wrong?
Wrong.
It's wrong. All body cells retain all the DNA actually, but some genes are expressed and some are not. Three, a small population of stem cells is retained in the body of adults throughout their lifetime. Is three right or wrong?
That's correct.
Of course, especially in the bone marrow, if you remember, the hemopiotic stem cells. Four, stem cells have more telomerase than other body cells, and this allows them to undergo an unlimited number of mitotic divisions. Is four right or wrong?
Um, right?
No. They have more telomeres. This is correct. But stem cells will never undergo unlimited number. They divide more than the normal cells, but not unlimited. Anything unlimited will be cancerous.
Oh, yes. Yeah. Okay. Yeah.
Okay. Unlimited means they never die. And every cell in your body dies at one point. So four is incorrect. So the answer is one and three. So the answer must be C. Okay. Good.
Yes.
Now, 23. Which statements are correct for all nucleotides? The nitrogen containing base is always attached to carbon atom 1 of the pentose. The phosphate group is always attached to carbon 5. A condensation reaction occurs to join the nitrogen containing base to the pentose. Nucleotides are linked together by condensation reactions between phosphate groups. Now, here, this question wants you to remember the structure of the um, deoxyribose.
One is
Now,
correct.
remind me. Excuse me?
One is correct.
Yes, one is correct.
Okay.
Two, the phosphate group is always attached to carbon number 5.
Yes.
Correct. Well
Okay.
done. Okay. And
Four
then
is
three, excuse me, a
four.
condensation reaction occurs to join the nitrogen containing base to the pentose.
Yes.
Yes. Well done.
And
It's
four is
one,
wrong
oh,
because it's phosphodiester.
are linked together by condensation reactions between phosphonucleotides are linked together by condensation reactions between phosphate groups. Okay, four is wrong. So 23 is A. Okay, 24. How many phosphodiester bonds are present in a circular DNA molecule of 2,700 base pairs?
2,700 times 2.
Yes, you are right. It will
So
be,
5,400.
yes, good. Okay, 25. Which statements about complementary base pairing are correct? Purines and pyrimidines are different sizes. Complementary base pairing occurs during translation. The base pairs are of different lengths, and uracil forms two hydrogen bonds with adenine.
Okay. Four is correct.
Yes, four is correct.
Yeah. One is correct to that.
Different side. Yes, one is correct. One has a single ring, one has a double ring. Now,
Two,
which one is
no?
wrong? Two or three?
Three, I think. But I'm not sure.
Three is wrong. Three is wrong. Because if you have base pairs, like if you have one nucleotide with a, okay, and another nucleotide with a base pair, complementary base pairing, the DNA has to have the same, the base pairs, the pairs, they have to be of the same length so that the DNA is constant throughout the whole length of the DNA. So the base pairs, they have to be of the pair, has to be of the same length at the end when they pair up. So three is incorrect.
Okay. Yeah.
Two is right.
So two.
Complementary base pairs occurs during translation. Yes. So it is one and two and four. 25 is B. Okay, Farouk?
Okay.
Good. Now, 26. During the mitotic cell cycle, the chromosomal DNA is replicated, and the specific points in DNA molecules where replication is occurring are known as replication forks. A typical human chromosome has about 150 million base pairs of DNA, and it takes about 1 hour to replicate the DNA of a typical human chromosome. The rate of replication using a single replication fork is approximately 50 base pairs per second. Approximately how many replication forks must occur in a typical human chromosome? Okay, how can we solve this question? We have to break it down. Do I help you or I give you time?
No, help me.
Excuse
Yes.
me?
Yeah. Can you help me, please?
Okay, now what would I do? I'll break this down piece by piece. During the mitotic cycle, the chromosomal DNA is replicated. Okay, very good. The specific points are known as replication forks. Okay, the typical human chromosome has about 150 million base pairs of DNA. So I will write here, this is the total number of DNA base pairs. And it takes about 1 hour to replicate the DNA of a typical human chromosome. So this is the total time. They're giving you some
Okay.
information. This sentence has to be important. Now, the rate of replication using a single replication fork is approximately 50 base pairs per second. So I'll write this information down. One replication fork will replicate 50 base pairs in 1 second. Okay. So how many base pairs will be replicated in 1 hour? 1 hour is how many seconds?
Uh, 1 second. Let me do the math.
1 hour is how many seconds?
3,600.
Yes. So we want to know how many base pairs will be replicated in 3,600 seconds. What will we do?
Oh, sorry. 50 times 3,600.
Yes, we will multiply 50 times 3,600. Then, 1 minute, let me calculate that.
It's 180,000.
Okay, good. So this is 180,000 base pairs will be replicated by one replication fork in 1 hour. Okay, now the question is, how many replication forks must occur in a typical human chromosome during DNA replication? So let me write this down here. We need one replication fork to replicate 180,000 base pairs in 1 hour. How many replication forks do we need to replicate the total number of base pairs, which is 150 million? That's six, yeah? 150. 1, 2, 3, 4, 5, 6. How many replication forks are needed to replicate 150 million base pairs of DNA? What do we do?
100,
150
150 divided by 180,000.
divided by 180. 1, 2, 3 equals. My answer is 833.3. So approximately how many replication forks? The closest answer is A.
Yes.
26A. Okay, Giandra, this is not a nice question. This is a tricky math
Yeah.
question. Okay. They are playing around here with 1 second, 1 hour. Okay. 27. Which molecule has its synthesis directly controlled by DNA? Amylase, cholesterol, glycogen, or phospholipid?
Amylase?
Well done. Yes, because it's an enzyme and it's a
Yeah.
protein. 28. Which statement correctly describes the association between a companion cell and its sieve tube cell? The companion cell provides all of the ATP used for energy requiring processes in both types of cell. The companion cell controls the active transport of molecules through the sieve plates. And the companion cell prevents site-to-site movement of assimilates between sieve tube cells. And the companion cell provides a nucleus that controls cellular activities in both types of cells. Now, this is very obvious. What's the answer?
Z.
D. Yes. 29. We have 10 more. Good. A maize seedling was grown in soil that contained lanthanum ions labeled with a chemical that fluoresces under ultraviolet light. The diagram represents what was observed when a section of the root was examined using a light microscope with ultraviolet illumination. So this is the fluorescent ion, and it will be absorbed by the root hair cell, and it will shine. These parts of the root hair cell will shine only. What does it show? What is the correct conclusion about the transport of the ions in the roots? The ions are not transported through the apoplastic pathway or the symplastic pathway. The ions are transported through the apoplastic pathway only. The ions are transported through the apoplastic pathway and symplastic pathway. I want you to answer based on what you see, not on what you studied, okay? The ions are transported through the symplastic pathway only. So what do you see in this investigation? How did the ions cross?
In the cell on the apoplastic, because the fluorescence is only in the cell walls.
Yes, the
So it's
ions
D.
are transported through the apoplastic pathway only because we only see the color and the cell walls. Very good. Well done, Farouk. Which description of adhesion and cohesion is correct? Adhesion refers to the force between the water molecules due to hydrogen bonding. Cohesion refers to the force between the water molecules and the xylem vessels. Is A right?
No.
No, A is wrong because cohesion is not between water and xylem. Adhesion refers to the reduced friction. Is B right?
No.
No, it's got nothing to do with friction. C, adhesion refers to the reduced friction again. No. D, adhesion refers to the force between the water and the xylem, and the cohesion refers to the force between the water molecules themselves due to hydrogen bonding. What is 30?
D.
D, well done. Excellent. 10 more to go. Which conditions are needed to allow the mass flow of sucrose in phloem sieve tubes? So the phloem sieve tube in the source and in the sinks. Can you read it and tell me which one is correct? Here we have water potential and hydrostatic pressure. Oh, I just gave you the answer.
It's fine. It was an easy question. Maybe.
Okay, this is easy because in the source, we have high hydrostatic pressure. So you might be confused between A and B, but it is high hydrostatic pressure and low water potential because we always describe it this way, that initially the water potential will be low because at the source, what is happening? We are loading sugar, sugar, sugar. So we will have low water potential. And this is going to take, Farouk, my dogs are barking 1 minute. I'll try to make them quiet.
It's fine.
Okay, let's go back. Yeah, in the source, we are loading sugar, loading sugar, loading sugar. So it will be very concentrated at the source, and we will have a low water potential. And because we have a low water potential, water is going to move out of the xylem by osmosis into the source, into the phloem. So this is going to make or increase the hydrostatic pressure at the source. I think you know this, yeah? So the answer is B. And the opposite happened at the sinks. At the sink, we are unloading, so the sugar is going out. It's getting stored. So we will have a high water potential. A lot of sugar is going out, out, out. So we'll have a high water potential. And because of that, water will move out of the sink into the xylem by osmosis. And then we will have a low hydrostatic pressure. You understand this? Yes? You understand why 31 is B?
Yes.
Okay, well done. Okay, 32. One type of congenital heart defect is where the left and the right atria are not completely separated. This is called the atrial septal defect. ACD usually results in blood moving from the left atrium to the right atrium. This causes increased blood pressure in the right atrium and decreased blood pressure in the left atrium. Which row describes other effects caused by the atrial septal defect? The blood pressure in the pulmonary artery will increase or decrease, and the blood pressure in the aorta, and the oxygenation of blood in the pulmonary artery. Now, think about it. Think about this question.
Okay, there is. So the blood pressure in the pulmonary artery will increase, I think.
Yes, you're right.
So the blood's gonna mix, the oxygenated and the deoxygenated blood is gonna mix. So the percentage oxidation will decrease, I think.
No.
No?
Look. An easy way to answer this question is to draw. I'll draw a heart if I'm you, and I'll divide it into the different chambers. Here is the left atrium, and here is the right atrium. I have the pulmonary artery coming from here, okay? And don't forget that the pulmonary artery is oxygenated or deoxygenated. Pulmonary artery oxygenated or deoxygenated? Hello?
Deoxygenated.
Yes. So they're telling you that the blood will move from the left atrium to the right atrium. And the blood in the left atrium is oxygenated or deoxygenated.
I'm sorry, Miss, can you say again?
I'm telling you that the blood is going to move from the left atrium to the right atrium. Okay?
Oh, so okay, yeah. It's the blood's gonna move to the pulmonary artery. It's oxygenated.
It's going to be oxygenated.
Mm.
So the blood is going to move from the left atrium. 1 minute, Farouk. 1 minute. Okay, I'm back. So they're telling you that the blood is going to move from the left atrium to the right atrium. The blood in the left atrium is oxygenated, okay? So it will enter the right atrium and it will pump from the right atrium to the right ventricle and from the right ventricle into the pulmonary artery. So now the pulmonary artery will have more blood because this blood came from the left atrium. So the blood pressure in the pulmonary artery will increase or decrease?
Increase.
Increase. So it is either C or D, okay? We will exclude A or B. Now the blood pressure in the aorta will increase or decrease? They actually
Decrease.
both decreased.
Yeah.
Are you with me, Farouk? Because
Yes.
blood moved from the left atrium to the right atrium. It should not do that, but this is what happened. Now here, this is the confusing part. The percentage oxygenation of blood in the artery, in the pulmonary artery, there will be more oxygen or less oxygen?
More oxygen.
Yes, because it came from the left atrium. So the answer for 32 is D. Did you understand that?
Yes, I do.
Pulmonary artery is deoxygenated, but now we will have more percentage oxygenation in the pulmonary artery because the blood came from the left atrium. Good?
Yes, good.
Okay, yes. 33. Now let me change color. Which property of water related to its role in blood and tissue fluid is correctly described? Water is the solvent for all biological molecules. Water is the solvent for most non-polar molecules. Water requires little energy to increase its temperature because it has a high specific heat capacity. Water cools down slowly because it has a high specific heat capacity.
Water cools down slowly because it has a
Yes, you're right. Well done. Okay, 34. Which statement about tissue fluid formation is correct? The hydrostatic pressure of blood decreases from the arteriole end to the venule end of a capillary. B. The water potential is always higher in the blood in the capillaries than in the tissue fluid and the surrounding tissues. No cells move out of the blood in the capillaries into the tissue fluid, or most tissue fluid formation occurs at the venule end of the capillary. Which answer is correct?
A.
I'm very happy. Yes, you're right, Farouk. Okay, can you tell me why A is right? It is correct.
Because the it decreases because the venue
It
because
decreases
it's tighter.
from the venule. Excuse me?
The blood decreases from the arterial end to the
Yes,
venule end of the capillary.
the hydrostatic pressure. We always used to draw it this way. We have the arteriole, and then we have the capillaries, and we have the blood cells, and then we have the venules that will join to make a vein. Okay, so here we have high hydrostatic pressure. That is why the tissue fluid goes out.
Yes.
Okay, and then the body cells will take the glucose and the oxygen and release the waste products. And then we have high hydrostatic pressure outside and the surrounding tissue fluid and low hydrostatic pressure inside. That's why the tissue fluid will move back into the venules, and the lowest hydrostatic pressure will be in the venule. So the highest hydrostatic pressure comes from always the artery or the arteriolar end pressure. Okay. And C is wrong. Why C is wrong? No cells move out of the blood. Yes, we know that the big proteins, they don't move out. But in times of infection, what will move out of the blood in the capillaries into the tissue fluid?
White, the white blood cells.
Correct. Well done. Oxyhemoglobin, carbaminohemoglobin, hemoglobinic acid, and carbonic anhydrase are found inside red blood cells. How many of these substances will show an overall decrease in the concentration as the red blood cell passes through capillaries in the lungs?
Okay.
I will not interfere.
Okay. Oxymoglin, carbinoxylin are not found. Not found. So overall, there's a concentration. That's fine. That's too clear. The the oxyhemoglobin will decrease. Wait, no, no, no. The oxyhemoglobin will increase. Sorry.
Yes, yes, we are at the lungs,
Yeah.
so there is
Yeah.
more oxygen, so the oxyhemoglobin will increase. Correct. What will happen to the rest, to the carbamino and the hemoglobinic acid and the carbonic anhydrase?
Yeah, the carbamino and the hemoglobinic acid will
Yeah,
decrease.
well done. And this is what?
This is an enzyme.
It's an enzyme. It's not going to be affected anyway. So, what are the two things that? Oh, I just gave you the answer. Oh my goodness.
It's B. So
What's the answer? It's B. Yeah. Okay. The carbamino hemoglobin and the hemoglobinic acid will decrease. Amazing. Which of these structures typically contain cartilage and cilia?
uh, bronchi and trachea.
Well done. Okay. Not the bronchioles. Bronchioles, no cilia. No, the bronchioles, no cartilage, no goblet cells. 37. Which statements about the function of tissues found in the human gas exchange system are correct? One, collagen in the bronchi prevents from collapsing, or smooth
No.
muscles in the bronchioles can contract to increase the flow of air into the alveoli. Elastic fibers in the alveoli stretch and recoil during breathing.
Okay, 3 is correct. Uh,
Excuse me, three is correct, of course. One and
yeah.
two here. Is one right or wrong?
No, one is not right.
Well done. Collagen is found in the blood vessels, not in the respiratory. We never mentioned collagen in anywhere in the gas exchange system. So one is wrong. One is wrong. This means I will exclude A. I will exclude B.
Yeah.
So what's the answer? And we said that, what's the answer?
D, because 2 is incorrect again.
Yes.
Yeah.
Muscles in the bronchioles, when they contract, they will not increase the flow of air, they will decrease the flow of air. So they're playing around with words. So it's three only. Well done. 38. Antibiotic resistant strains of whatever are a major problem when treating TB. A new antibiotic, Teixobactin, could be very effective at killing the M. tuberculosis with only a small risk that the bacteria will evolve resistance. Penicillin and similar antibiotics bind to a single protein, but the Teixobactin binds to two lipids that are needed for the formation of the bacterial cell wall. Teixobactin binds to regions of the two lipids that do not vary across many different species of bacteria, which statements help to explain why the use of the Teixobactin is thought to be less likely to lead to the evolution of antibiotic resistance than the use of many other antibiotics such as penicillin. Okay. A single mutation can result in bacteria that are resistant to penicillin and similar antibiotics, but at least two mutations are required to produce the Teixobactin resistant bacteria. Let's stop. One is right or wrong. If you look at this sentence, they're telling you that the Teixobactin, this antibiotic, it binds to two lipids that are needed for the formation of the bacterial cell wall. Is one right or wrong?
That's right.
Yes. I need here at least two mutations to produce the resistance strain because we need to affect two lipids. Two, mutations can affect the structure of proteins but cannot affect the structure of lipids because only proteins are made of amino acids. Now, is two right or wrong?
No, it's incorrect. It can
Of
affect
course,
the.
it's incorrect. You're right. It's incorrect because a mutation can affect anything. What if enzymes are needed in the making of those lipids? And these
Yeah,
enzymes are mutated, so the lipids will not be made. So two is wrong. Once I am sure that two is wrong, a very easy way is to exclude any answer that has two. So I will exclude A, I will exclude B, I will exclude D. So the only answer left will be C. And let's look at number three. It should be right. The lack of variation across many species of bacteria in the two lipids that bind to the Teixobactin suggests that the particular structure of these lipids is essential for the bacterial cell wall formation. Didn't they say that already?
yeah, I think.
Here, it's needed for the formation of the bacterial cell wall. So three is very correct. So it is one and three. Okay. 39. Monoclonal antibodies are now being used to treat some human diseases. What explains why monoclonal antibodies are suitable for this purpose? Now read and tell me.
Okay. They can divide by mitosis to produce the large numbers of antibodies required for the treatment. No, because you can. Yeah.
One is right or wrong?
No, one is wrong.
Correct.
Because that's not specific to.
So I will remove A and I will remove B because monoclonal antibodies, they don't divide by mitosis. It's the plasma cells or the memory cells that divide by mitosis. These are not cells to divide, they are antibodies. So one is wrong. Two, they are specific to a particular antigen and they can be modified so that they do not act as antigens themselves.
Yes, they're both correct.
Yes, both are correct. They are humanized, if you remember. Good. The last question. Measles is an infectious disease caused by a virus. The graph shows the number of cases of measles each year in a country before and after a vaccine was introduced. So the vaccine is introduced here, and then obviously there is a decrease in the number of cases of measles. What could have caused the decrease in the number of cases of measles after vaccination was introduced? Vaccines, do they
Vaccine.
provide artificial active immunity?
Yes.
Yes. So it's not B. The easiest way is to exclude, not B, not C. It's A or D. So choose A or D.
Okay. A, because fewer people are able to act as hosts for the virus.
And also, vaccines provide artificial passive immunity. No, correct. Okay, so the answer is A. Well done, Farouk. You did a very good job.
Thank you.
I think the exam was like average, not very easy, not very hard. Okay, I'll save that and send it on the group
Yeah, would you
and
mind
meet
sending a
you.
blank copy as well, miss?
Excuse me?
Would you mind sending the blank copy, like one
Yeah,
without?
I'll send both. Okay. I'll send the blank coffee and I'll send the answer card. Okay.
Okay, thank you.
Anytime you have a question, send me. Okay, welcome, my dear.
Thank you so much.
Bye bye.